Hi everyone, welcome to week 4. You are all doing so well, and I hope that you feel like you're learning something, and I also hope you feel like you are more than able and welcome to reach out to both myself and Dr. Wilburn if you are having questions and need some clarification. I promise it's not just you, so reach out and usually we can help clarify some things within 5-10 minutes, and then you're going to be better off for the rest of the semester for having done that. So again, please take us up on that help. The devotion for today is from Matthew 25:34-40. Then the king will say to those on his right, "Come, you who are blessed by my Father, take your inheritance, the kingdom prepared for you since the creation of the world. For I was hungry, and you gave me something to eat. I was thirsty, and you gave me something to drink. I was a stranger, and you invited me in. I needed clothes, and you clothed me. I was sick, and you looked after me. I was in prison, and you came to visit me." Then the righteous will answer him, "Lord, when did we see you hungry and feed you, or thirsty and give you something to drink? When did we see you a stranger and invite you in, or needing clothes and clothe you? When did we see you sick or in prison and go to visit you?" The king will reply, "I tell you the truth, whatever you did for one of the least of these my brothers, you did for me." I think this is very pertinent and very important always, but especially in these times. I truly believe that God created all of us and God loves all of us, and therefore God, Jesus, the Holy Spirit are in each and every one of us. If you saw someone walking down the road and you knew that person was Jesus incarnate, how would you treat them? Well, I truly feel that we should be treating each other the exact same way. Each of us are loved, created, and a part of Christ's kingdom, and if we remember that, hopefully we are treating them that way, and as Christ asks us to treat each other in this. So, not always easy, but something important to remember. All right, with that, let's go on. This chapter 4, is all kind of about cell organization. So organelles, how the cells work. And so what I want you to get from this video, and then some of the things we talk about on Wednesday, is the general properties of cells. So what is required for cellular life? All right, we'll talk a little bit about organelles and cell organization, but that's something you're going to learn largely on your own through the organelle matrix homework. I want to talk a little bit about properties that organize the cell. So if you think of the cell as being a very active, organized entity, almost like a city where there's different parts of the city that have different purposes, that city needs to be organized, and the cell also needs to be organized. The proteins need to get to the right place, they need to have the right function, just like doctors need to be at the hospital to do their job. Garbage collectors need to do their job in the right place. Garbage collecting is extremely important, but if garbage collectors are collecting the wrong things or doing the right job in the wrong way, that's going to be a huge problem. So the cell has to be organized. How does that happen? How do cellular systems make these properties possible? So we're talking about kind of how all of these things work together in the cell. So it's one thing to know about each organelle, but how do they work together to make all of life possible? And so this includes, again, getting proteins to the right place, and then kind of, again, what does each of those organelles do? And this is going to be mostly on your own, kind of as part of the homework. And so the way I like to think about this is the idea of reduction biology and systems biology. So reduction biology is what we're doing a lot in this course. We're really reducing life of the cell down to its most basic entity. So we're really going to learn about those macromolecules we've been talking about. What do the proteins do? What do the nucleic acids do? The lipids, carbohydrates. And this is kind of, in this chapter, we're also learning about individual parts of a cell, like the membrane and the organelles. What are they doing? And this is really important because if you want to understand disease, for example, if you want to understand a mitochondrial disease or a disease of the respiratory system, you really need to understand what's happening at the protein and enzymatic level. What's happening that causes that person not to make energy efficiently? And therefore, that understanding will help you not only diagnose the disease, but know how to treat it and so forth. So biology is really important to kind of reduce it down and understand the mechanism of what's going on in all these things, right? But at the same time, if you just understand that particular enzyme and what that's doing and don't put it back into the big picture of what that does with the disease or the tissues as a whole or that whole individual, then you're also missing something as well. So you need to be able to put those two aspects together. What's happening at this macromolecule, the mechanism level, but how does that work together with the rest of the cell, with the rest of the tissue, and the rest of the body? And so with that, those interactions of those components, each protein is important, but it's the way that protein, that organelle, that cell interacts with everything else in the body that ultimately will make it. And so I kind of think about this like a puzzle. If you're going to put together a puzzle, it is important to understand and see the characteristics of each piece of the puzzle, right? But unless you actually put that together and see the whole picture as well, you're really missing key aspects. So the details of this puzzle piece are really important, especially if you live in Mexico. That would be right here. But if you don't put it together with the whole picture, you really have a hard time seeing what that means. Right? So with that, let's kind of move forward. Let's talk about the cell theory. So go ahead and just on the question, see if you can pick out what components of the cell theory are there. All right, so the cell theory basically states that all living organisms are composed of one or more cells. Cells are the smallest units of living organisms, so these two things are very much related. If all living things are composed of at least one cell, that means cells are the smallest units of those living organisms. And then finally, and this is really important, all cells come from other cells. All right? And so why is this? How can we think about this a little bit more? Well, what's required to make a cell? All right, so I'm going to give you the first one, a genome. We're going to talk more about DNA, not only in this chapter, but throughout the rest of the course. But you need a genome to basically have the blueprint for that organism. So let's say for a second that I was an amazing biologist and I was able to get the complete set of DNA from E. coli. Even a bacteria has complex DNA. So about the simplest set of DNA, still complex, and I put it in a test tube, am I going to have a cell? Well, the answer is no, because the genome cannot work on its own. We'll learn later on in this semester about RNA polymerase. So RNA polymerase is needed to basically read that DNA and create a messenger RNA from it. And that messenger RNA will then be needed to go forth and make proteins. So we have to have ribosomes, and ribosomes read that mRNA to make proteins. So if we just had a genome and we had no RNA polymerase already there, and we had no ribosomes already there, and tRNAs, and all these machinery to actually take that genome and turn it into the proteins needed to make that cell work, well, the genome is just that, a genome. It can't become life as a cell. All right? So what else might we need, though? So let me ask the next thing. So let's say I am able to put the genome and those pre-existing molecules. So now I have all my ribosomes, the RNA polymerase, all those key protein enzymes. So I can take that genome and I can use it to start making all the proteins that are required to basically make the cell function. Do I have a cell now? What do you think? Why or why not? Well, the answer is no, because I still don't have the structure that's required of a cell. I need a membrane system, for example. I need pre-existing organelles. And so all of these things are really important to make a cell as well. And so since I need a genome, pre-existing molecules, and pre-existing cellular structure, that means that I need a pre-existing cell to make a new cell. And this is why reproduction works the way it does. So the sperm, basically, if you're human, you have 46 total chromosomes, 23 from dad, 23 from mom. The sperm brings in its 23 chromosomes from dad, and then the egg from mom has their own 23 chromosomes. But the egg provides so much more as well. The egg provides those pre-existing molecules. There's already RNA polymerase, ribosomes, and things ready to go to help make the proteins as that baby starts to develop. And that egg already has membrane system. It already has that cellular structure. We could not make the new organism if we didn't have the pre-existing cell, which in this case is the egg. And so upon fertilization, that organism, that baby, starts to grow, and from there all of the new cells divide and undergo mitosis. So all cells come from pre-existing cells. Let's talk about some other properties of cells. Well, all cells have to acquire and utilize energy, and we'll learn all about this in Chapter 6. But this is the aspect of metabolism. So catabolic reactions are going to break down things to create energy, and then anabolic reactions build up things. So when we eat, we're eating proteins, we're eating nucleotides, we're eating all kinds of things, because usually we're eating something, well, hopefully we're eating something that was alive, whether it was alive as an animal or alive as a plant. But in doing that, we're going to break down, digest those things, and by breaking them down, we're going to get energy. And we're also going to get the building blocks. This is like tearing down a building to get the individual bricks, so that then we can use those bricks to make the type of building that we want. We're breaking down the DNA, the proteins from our food, so that we can make the DNA proteins that we need, that our cells and our genome require. In doing so, we get energy breaking down, and we need energy, use that energy to build up. Also, cells are dynamic. Cells are constantly moving, right? So there's a few examples of this. So here we just have cells growing in culture. These are monkey cells, and you can see this is a time lapse, so this isn't real time, but it's not too long. These cells are moving. Cells are constantly moving, and even cells in your body are constantly in motion and reacting to the environment and so forth. We also have movement inside the cells. You may have seen this video before. I know that in my class at least we watched this on the first day. And this is a motor protein that's basically walking along this microtubule, another organelle that you're going to learn about in this chapter, and it's pulling a vesicle. So we're actually going to talk about vesicles and how they're used to get proteins to the right place for certain parts of the organelles. But this is things moving inside. This is almost like a transport truck moving something along a road to get things to the right part of the city. Well, this is a motor protein pulling a vesicle, excuse me, with proteins or cargo inside it to get to the right place. So there's things constantly moving. The cells are dynamic. And then that's not to mention the thousands and thousands of chemical reactions that are ongoing in the cell all the time. And these are always changing depending on what the cell needs. And so how does the cell know what it needs? Well, it has to respond to stimuli. This is going to be Chapter 8. We're going to talk in Chapter 8 about how the cell responds to extracellular molecules and to signals from around it, and those signals are then interpreted by the cell so that the cell changes what it's doing. It changes if it's moving or not. It changes what types of reactions, chemical reactions it's undergoing, and so forth. So just to give you an example of cell movement and a cell responding to the environment, this video here is going to show a neutrophil. So a neutrophil is one of your white blood cells, so it's one of your immune systems, and it's actually chasing this bacteria up here. And the bacteria looks like it's running away from the neutrophil, but it's really just doing random motion like bacteria do. But this neutrophil is basically reacting to protein and kind of debris that the bacteria leaves off. So we are going to learn, this is Chapter 5, we're going to learn about all the things that are on a membrane. And so a lot of those proteins and carbohydrates and things like that fall off. And they're going to bind to the neutrophil, and the neutrophil then basically knows which side of it that bacteria is on, and it really kind of responds and looks like it's chasing it. So I think this is pretty cool. So here we have the bacteria. Again, this is time-lapse, but there's the neutrophil chasing it, and the bacteria, wherever it goes, the neutrophil kind of reacts and changes its motion along the way until eventually it's finally be able to surround that bacteria and engulf it, and in doing so basically protecting you and your body from infection. Alright, so that's an example of a cell responding to stimuli and in doing so regulating itself. But there are other things a cell must do and must regulate. For example, it needs to regulate division and growth. We're going to talk about this in later chapters as well. A cell should divide, but it needs to divide when it's supposed to. It needs to replace dead cells. We need to grow, right? But it also shouldn't divide when it's not supposed to. When a cell's dividing when it's not supposed to, we all know what that causes. That causes cancer. And so obviously, or hopefully fairly obviously, cell growth occurs at different rates at different points in your life. And my favorite biological example is my son Ethan. So he's actually 15 now, but this is a picture of him when he was 4, and we took him to Disneyland. And at birth, Ethan was 9.5 pounds, 20 inches. So actually quite a big baby, nice healthy baby. Too big for my wife Beth's liking, but anyway, nice and healthy, he's doing great. By age 4 at this picture, he was 40 pounds and 42 inches. So essentially what that means is in 4 years, his weight had quadrupled and his height had basically doubled. So if he continued at that growth rate, assuming you all are somewhere around 20 years, by your age, if his weight continued to quadruple every 4 years, he would be 9,728 pounds at age 20, and he would be 53 feet 4 inches tall by age 20. So obviously, his growth rate has slowed down as he continues to get older, and that's not even to talk about the growth rate in utero before the baby's born. So he's actually now 15. This slide, I guess, is a year old. But at age 14, if he hadn't already slowed down, he would already be 1,200 pounds and more than 18, almost 19 feet tall. So while he has grown a lot, he definitely is not quite that big. So cells do regulate their growth, their activities, and so forth. They also have to regulate the metabolism. There's times when your cells need more energy. For example, when you're working out, your muscles need more energy than when you're just sitting down or sleeping, watching a movie, whatever. Regulation of gene expression, we'll talk a lot more about this in the second half of this video. And then finally, undergoing differentiation. We'll also kind of talk about this. But when you are first conceived, the sperm binds to the egg. That stem cell basically has to become every cell in your body. But as you become more and more cells, they have to start differentiating and becoming specialized. Your muscle cells have a different role than your neurons, which have a different role than your red blood cells, and so forth. Right? And that's the process of differentiation. And all of this really starts, again, with the genome. The genome basically is the blueprint for your body, and the genome is kind of guides a big aspect of who you are physically and probably other things as well. All right, so I'm going to pause here for a second, and we're going to change gears just a little bit, and we're going to move on into the whole idea of genome versus proteome in a minute. But one of the things that you are supposed to learn, or we want you to learn in this chapter, is organelles. All right, and so figures 4.8, 4.9, and 4.11 are really important and really good, kind of talking about the organelles and where they are in the cell and so forth. All right, but I want to do a quick preview of this, so let's ask a few questions. You've got the options: cytosol, nucleus, endomembrane system, and semi-autonomous organelles. And you can see in these cells, at least the animal and plant cell, remember prokaryotes don't have organelles. In these cells here, you have them labeled green, yellow, kind of purple, and blue. So on your Edpuzzle questions, go ahead and answer, what is the color code of these diagrams? So what is the green representing? What is the purple and pink representing? All right, and here's your options again. Get my picture out of the way. What is the blue representing? And what's the yellow representing? See what you come up with. All right, hopefully that went okay. The organelle matrix is also going to guide you through this because I really want you to understand the main function of each organelle. That's probably the most important aspect. What does the lysosome do? What's its function in the cell? What's the mitochondria do? And so forth. But in order to understand that, you also need to understand a few key components. What is that organelle made up of? And does it have its own membrane or not? And so these are all parts of the organelle matrix. This is something that I really think you can do on your own. Between the textbook information, I've linked a really good YouTube video on Canvas that you can watch for this information, and there's all kinds of information on the internet about each organelle. I think you can basically come up with that, and but always bring us questions, and we'll review them very briefly on Wednesday anyway. But if you have questions, ask us, ask your TA, get the information you need. All right, so let's jump to genome versus proteome. So I'm going to ask a few questions along the way here. Remember that these are just thought questions. Do the best you can, and then we'll talk about them. So what do you think is the best definition of a genome? All right, genome refers to the DNA. All right, so chromosomes have a lot of DNA on it, but a chromosome would not be your entire genome, because remember, as humans, we have 46 chromosomes. All the genes on that chromosome still does not encompass all the DNA or all the genes that make up your body. You need to talk about all the genes on all 46 chromosomes. So now we're talking about the entire collection of DNA, all the genes, all the chromosomes that are the blueprint for the building of a particular organism. So we're talking about this one here as the correct answer. Why isn't it the entire collection of nucleotides within a cell? Well, remember from chapter 3, we don't just have DNA in our cell, we have a lot of RNA. And that RNA is really important, has major functions, but that's not a part of the genome. So that's why this wouldn't be correct, because the entire collection of nucleotides includes that RNA that's actually being made from the genome. And then the entire collection of genes and proteins, well, proteins are what the genes code for. Genes code for some other things as well, but the proteins, again, are what the genes code for, and so that's beyond the genome as well. All right, so what's the best definition of a proteome then? All right, anything that degrades proteins would be called a protease, ace degrading, and so microorganisms that degrade that really have nothing to do with the proteome here. An organelle for protein synthesis, again, kind of not the proteome. If the genome is the entire set of genes in your cell, the proteome would be the complete set of proteins in that cell. All right, so I'm going to come back to why E is incorrect in a second, but first I want to move on to this next question. So which of the following changes throughout the life of a cell? Is it the genome? Is it the genetic code, the proteome, the number of copies of any particular gene within the cell, or the cell's hair color from brunette to dyed blonde to red, white, and blue, patriotic phase, to gray, old, or blue, very old? Sometimes I write these questions very late at night, so I like to think I'm funny, so I'm just imagining you laughing hysterically right now, and I'll keep that going. So your kind chuckle is appreciated. But go ahead and answer the question. All right, this is probably the biggest key here. The proteome is what's going to change throughout the life cycle of a cell. Remember, the proteome is the set of proteins, the complete set of proteins in a cell at any given time, right? So, but the proteins that the cell is making is going to depend on the environment and what the cell needs. So as the cell reacts to the environment, it is constantly changing what its proteome is, so what proteins it's making and what the proteome is doing. And this is true based on reaction to the environment, what the cell needs for your body, but also during differentiation. So for example, as that cell starts to differentiate into a muscle cell or a neuron or so forth, the genes that it's using, the proteins that it's making, is going to constantly change. So the genome, and this is absolutely key, the genome of your cell stays the same. The blueprint of your cell is staying the same. But the proteins that are made from that genome, those are constantly changing in response to the cell's needs. So the proteome is constantly changing, the genome is constantly the same. And this is why the proteome is not the complete set of proteins a cell can make throughout its lifetime, because the capability of making proteins is based on that genome. So all cells have the exact same genome, all cells have the exact same blueprint to make the exact same set of cells, but the genes that are used, the proteins that are made, are going to be different between different cells and are going to be different based on that cell's needs throughout its life. So again, the proteome is changing constantly, all right? And this leads us into kind of the last important concept that I want to get to. So how do the differences in the proteome of a muscle cell and a nerve cell, for example, come about, right? So what is making, our red blood cells, our nerves, all of the different cells in our body act differently? Because they have the exact same genome, but they are very, very different. They have the exact same blueprint, but they're very different. Right? So see what you can come up with, brainstorm, answer as best as you can, and then we're really going to get to this. This is the last concept for this part. All right, so let's talk about this together. So to answer this, I'm actually going to start with another question. So what's at the basis of the differences between different species? So here we have a phylogenetic tree, and you'll learn all about phylogenetics and everything in Bio 2011 next semester. But what is the basis for the difference between all these different organisms? Right? And the difference is differences in the genome. The genome of these different organisms is different, hence they are different. So if the blueprint is different, you're going to make something that is different. And so I have two analogies for this. Right? One is what I've been talking about before. These are the blueprints for the body. So if you take the idea of blueprints with buildings, you can have two different sets of blueprints, and when those instructions are different, you're going to make a different building. The building's going to look different, it's going to behave different. So if you have different genomes for two different organisms, that blueprint for the body is going to make that organism look different, behave different, and so forth. Right? Now, within the blueprints, there may be a lot of similarities. For example, both blueprints might really talk about electrical wiring and construction and use the same terms like hammering and all those other aspects. So the blueprints may be very similar, but the difference in the instructions leads to the difference in the buildings. Right? And that's true for the body too. The instructions, a lot of the things that are there are going to be extremely similar between different organisms, but the sequencing of the genome, very slight differences, can make a big difference in what the body looks like and how the organism behaves and acts and so forth. The other analogy that I'm going to carry a little bit forward is the analogy of cookbooks. Right? So let's think about this. So here we have 3 different cookbooks. Right? So the letters are the same. Right? Just like the genomes of all different organisms, all organisms have A, C, T, and G in their genome, and then U is added for RNA. So if assuming that these are all in English, the letters are all the same, A, B, C, D, and so forth. However, they're arranged in different ways, and the different organization or the different arrangement of those letters means something completely different, right? GOD means something different than DOG, and both of those mean something different than CAT. So if we use letters and we rearrange them, we have different meanings, right? And so the difference in the genomes between 2 different organisms is kind of like different cookbooks, written in the same alphabet, but they have different meanings, different recipes. Now, within those recipes, you might have a lot of similarities. You know, both of these recipes might include butter, or you might have recipes that have sugar, and they might include terms of like tablespoons or half cups and things like that. So a lot of the aspects may be extremely similar, but the instructions are a bit different. And that's kind of what allows us, the different organisms, to be different. It's the differences in those genomes. But those similarities are still extensive. Humans and chimps have 3 billion base pairs in their genomes, but there's only 40 million differences. 40 million might sound like a lot, but amongst 3 billion, that is not a lot. So basically, humans and chimps are between 98 and 99% exact sequence of the genome. So in other words, our 2 cookbooks are 99% the same, but that 1% difference really makes a difference in the body structure and the difference between chimps and humans. Even mice and humans are about 85% similar. And so, but still, the genomes are different, therefore the cookbooks are different. All right, so let's jump to the next thing. We know, though, in our body, within our body, we have 10 trillion cells. Each one of those cells has the exact same genome. Give or take, there can be a few mutations that happen later on in life. But basically, they have the exact same genome. And we know this because they all started from that single cell where the sperm fertilized the egg. And then that cell divided, and the genome was copied, until you got 10 trillion cells. The genomes are exactly the same. And yet the cells look and behave and have such different functions. So how can that be? Well, go ahead and think about it and answer as well. The reason relates to the choice of which genes are used. We have the genome for over 20,000 genes, but not every cell is going to use the same combination of those 20,000 genes. There are some genes that are more useful for a neuron, and there are other genes that are more useful for a red blood cell. So only certain genes in each type of cell are active, and the genes that are active are different for each cell. And so, continuing on that cookbook analogy, I may have this chocolate cookbook, you may have the exact same chocolate cookbook, but it doesn't mean we're going to make the exact same recipe tonight. Or if we have a broader cookbook, if you and I have the exact same cookbook, it doesn't mean we're having the exact same dinner. I may choose one recipe for my main course and a different recipe for my side dish and a different recipe for dessert. And you may have the exact same cookbook but choose 3 different recipes. Or maybe your main dish is the same as mine but your side dish and your dessert is different. So just because we have the exact same recipe book doesn't mean we're having the exact same dinner. The difference is in the choice and the combination of choices of which recipes we use. So just like that, the differences in these cells are the differences in which genes and which combinations of genes are used. So let's think about this one last way. The fertilized egg, this is way overly simplified, but let's just say we have these three genes. We have myosin. Myosin is the motor cell that helps muscles move. So myosin is needed for muscles. Hemoglobin is really important for carrying oxygen in a red blood cell. And acetylcholine is a neurotransmitter, so obviously nerve cells need acetylcholine. Every cell in your body has the gene for myosin, hemoglobin, and acetylcholine. But when that cell differentiates to a red blood cell, that gene for myosin is going to be turned on and used, and a lot of myosin is going to be made because that gene is being used frequently. However, the muscle cell doesn't need hemoglobin or acetylcholine. It still has those genes, but they're turned off in the muscle cell. In contrast, the red blood cell doesn't need myosin, but it sure needs to make a lot of hemoglobin. So in these cells, that gene is turned on, and the proteins for hemoglobin are made at high levels, and acetylcholine is not. Whereas in the nerve cells still have the gene for myosin and hemoglobin, but don't turn them on. They're turning on the gene for acetylcholine at high levels. So the genomes of all three of these cells are exactly the same, but the proteomes are different, depending on which genes are turned on and which ones are turned off in each. And that difference in the usage of the genes, the difference that makes the proteomes different between all these different cells, gives them their unique shape, structure, and function in your body. All right? So, in summary, how do the differences in the proteome of a muscle cell and a nerve cell come about? Well, it's the differences in which genes are turned on. It's the differences in how strongly those genes are turned on. So two different cells may actually be using the same gene, but at different levels. One type of cell might be using a lot of that gene, and another cell just a little bit. We'll get into the details of some of these other aspects later, but even within the same gene, you can make different proteins. So later on in the semester, we'll talk about alternative splicing. So the same gene can actually be used to make different versions of a protein, and so that can make cells different as well. So even though we only have 20 to 25,000 genes, we might make about 100,000 different proteins based on how those are cut. And then finally, which proteins are active and which ones aren't. This really is something that you'll get into in molecular biology, and if you take developmental biology, this is really important. But even when the proteins are made, the cell can control their activity by turning some on and some off at different levels. So this is the exact important aspect. Every cell in your body has the exact same genome, but they behave differently because they have different proteomes. Hopefully that makes sense. As always, come to us if you have questions. Good job.